Hello again, and welcome to the Crop Protection Network Spring Webinar Series. We are going to go ahead and get started. This series is hosted by the Southern IPM Center with funding from USDA National Institute of Food and Agriculture, otherwise known as NIFA, the Crop Protection and Pest Management Regional Coordination Program. We are one of 4 regional IPM centers supported by USDA NIFA to coordinate IPM across the region. I am Kayla Watson, the Communication Director for the Southern IPM Center, and this is the first presentation in the spring series. So today we're going to be hearing from Marty Chilvers, who's an Associate Professor at Michigan State University. And if you have any questions as we go throughout the webinar today, you can put them right into the Q&A box, and we'll be holding those questions till the end. So you can see that on your Zoom panel right at the bottom of your Zoom screen. So Marty Chilvers earned his Bachelor's of Agricultural Science degree with honors and PhD from the University of Tasmania, Australia, with subsequent postdoctoral training at Washington State University. Dr. Chilvers began his position with Michigan State University in 2008, where he leads a field crop pathology program researching plant diseases and management solutions. So the presentation that he'll be giving today is an update on soybean seedling disease research and management. So welcome, Marty, and thanks for giving us this presentation.
Thanks, Kayla. Thank you very much for the introduction. My audio level okay?
Sounds great.
Excellent. Thank you. Yeah, and if you have any questions as we go through this, please pop them in that Q&A box and we'll try and get those addressed. Okay, let me get this presentation started here. All right, okay, you should be seeing my slide deck now. So, yeah, so I'm at Michigan State University then. This is some work across different grants. So USB, local Michigan soy, NTSFP, so industry funded projects too, which I don't have the logos up there, but let's get started, I guess, and I'll jump into what we're going to talk about. So today I'm not really going to be doing a straight up sort of comparison shootout of products or anything like that. What I'm sort of trying to do is give you maybe a behind the scenes sort of look at what we do in terms of trying to understand, the cause of seedling disease so that we can improve management options. There's a lot of, partnership with industry there to try and figure out different products and with breeders in terms of germplasm. And then ultimately what we want to do here is we're working towards risk prediction systems, especially, I guess, given some of the concerns that we've had in a regulatory sense with neonics and other sorts of seed treatments. If there's any way of actually predicting risk to know where we need to place these things, then that might be very, very helpful. And, hopefully towards prescription management, and certainly some of those options are becoming more viable now with in-furrow treatments that are gaining some popularity and those sorts of things. So the 4 different sort of sections or things that we've done work on in the lab is some seed treatment return on investment work. We pretty much do these types of trials every year. I'll focus on one in particular across the state. An seed survey that was a national one, some phytophthora work that was back here in Michigan, and some soybean sudden death syndrome work that's also going national now. So the first one I'll just talk about here is a soybean seed treatment profitability trials. We ran this for a few years, and Devin Rossman was the master's student that worked on this. We picked a couple of Asgrow, a couple of Pioneer varieties, and went with the standard seed treatments that were available to growers. So no treatment at all, so an untreated check, fungicide only, fungicide, insecticide, all the what you would consider the full package with the Poncho Votivo. And then so the first year, I thought this is going to be great, super exciting, pretty clear results that, the seed treatments are absolutely going to be critical by the looks of it, right? So these are color coded and it's yield relative to the untreated check. So you can see that most of these instances, all the averages are positive. And there's particularly one location here on the west side of the state in Allegan County that was very much positive, right? So we had some heavy rainfall after planting, and we saw, up to 10, 15, 17 bushel protection compared to the untreated check. This was at a higher seeding rate, 160,000 seeds per acre, and planted on about par with most of where Michigan soybean seed gets into the ground. So there's certainly caveats to any data set, and I would say that the planting rate is certainly one of those. But it was interesting to see these different responses, and I thought, okay, this is really good first year study. We'll see what comes in the next year. And just before I transition off this slide too, one of the other things that was really curious here is differential responses by variety. So there's an interaction between variety and seed treatment. So we can see that here at this location, Hillsdale. One of those locations is providing us around about a 5 bushel protection, and the other one is pretty flat. And again, here at that Allegan, that highly sort of disease rich sort of environment, you've got differences across those two different varieties of one of those, performing much better than the other and being statistically different in this particular instance. So we thought that was really interesting, and we conducted that for a few more years. I'm not going to go through all of the different locations and years and the ways we can break that out. But here's a piano chart just to sort of break that out. And this is looking at the full package there across the 3 years and across those 7 site years that we had each year, so 21 different site years. And yeah, unfortunately, it's not always super, always that positive response, right? If it was, it'd make my job a lot easier in terms of making that recommendation. Yes, you should definitely do this because you're always going to get a return on investment. There's certainly that insurance perspective as well, component to this, and uniformity across fields, wet areas and whatnot, so different stresses in different parts of the field. But just on average, this is the split we were seeing. And on average, we're only seeing about a 0.6 bushel response across all those different site years. So certainly some pretty big variability there across those site years. So just to sort of summarize that, and this is sort of leading into why we're doing some of the other work we're doing as well, like trying to figure out exactly, okay, that's great, we've got these variations, but what is going on? What is driving that? What are the pathogens there? What are their fungicide sensitivities? Which treatment should we be using? And so on. So we pretty much always saw that some of these locations like Allegan were very responsive to that seed treatment. So there's something in particular going on there. There's certainly nothing that clearly jumps out as an immediate risk predictor, and that's what we can run with and make risk predictions across many other locations. There's a lot of variable factors, but it was super interesting, and that's certainly something we see when we do any sequencing type projects. If we do any sort of what we call amplicon sequencing, where we extract DNA and then sequence to see who is present in that soil sample, we see the biggest diversity by location. So each location tends to have a somewhat different community versus within a location. And so from this work, certainly, those seed treatments are going to obviously increase return on investments when we're planting early into higher stress sort of conditions, and certainly at lower planting populations as well. And there's certainly been, other studies, something that Sean Conley put together looking at neonics, and I think it was in the range of half a bushel, maybe a little bit more on average. So, maybe some question there as to the benefits. So what causes seedling diseases? There's sort of 4 main categories, and we can subdivide those into 2 larger groups. So we've got the oomycetes, the Pythium and phytophthora that produce a free-swimming zoospore, and then the fungi that don't need that free water. I mean, I think they certainly benefit from that. We see it with rhizoc and fusarium too. We get some saturated soil conditions. That can be certainly very favorable for infection, but it's not as necessary as it is for the oomycetes that have that motile phase of infection. But a lot of states don't really know who the players are, and so this is what sort of spurred some of this work that was both NIFA, USB, and NCSOP funded. And there's some work, I'm going to focus right now on the oomycetes and the work we did, but there's also some work that was done on the fungal species as well. So we did this big survey effort out across the soybean production region, as far south as Arkansas and up into North Dakota and across the border into Ontario as well. And we targeted fields that had a known history of disease or that had emerged and appeared to have some sort of stand issue. And so from each of those different locations, all of our collaborators, our other university collaborators in these other states, collected isolates and then sent them to Michigan for identification. And again, focusing on the oomycetes that were present. And this is what we found over 2 years. And this is a pretty simple graphic of about 3,300 isolates, but it was an incredible effort from all the collaborators to collect all of those isolates and ship them down. A single slide really doesn't do it justice, but it was really interesting what we found. So this immediately gave us some idea of which species were there, how they varied by year. And then we could start asking questions. Okay, so we find all these different species, which ones are really causing disease? Because just if you isolate something from a disease plant doesn't mean that it's necessarily the pathogen. It could be a secondary organism, it could be an endophyte, there could be other reasons why it's associated with that plant sample. So just looking at the top of this graphic here, Pythium sylvaticum was the most abundant across both years, and this is looking at 2011 compared to 2012. Pythium upapillum, which not much work had been done with that particular Pythium species, and I don't think it was reported as a pathogen at that time either, was the second most abundant in 2011, but that really dropped off in 2012. And if you remember back that far, 2012 was a very dry, warm season, right? We had a lot of drought conditions in many parts of the country. And so that may very well have been driving these differences by season. And certainly we start picking up on that as well when we start looking at more of the pathogenicity data that I'll show you in a minute and some of the different temperatures that we apply to these different species. Just for reference to Phytophthora sojae was down here, fairly low frequency, but it was present as well. So all up, I think we had about 82 different oomycete species in total represented here. So this represents a fantastic collection too to now ask, okay, which ones are really pathogenic? And then if they are, what's their fungicide sensitivity level? Like what products can we use to control them? So we set about asking which ones were pathogenic using two different types of assays. One we call like a seed rot assay. You can see here's a control plate. We've got good growth, good germination of that soybean seed. And then some other examples here, Pythium irregulare, quite aggressive, preventing a lot of that seed from even germinating. Pythium sylvaticum, that most abundant species, and Phytophthora sojae. So interestingly, Phytophthora sojae was doing a little bit of damage, but it certainly was nowhere near as aggressive as these Pythium species in these seed rot assays. And the other type of assay that we had was a seedling root rot assay. So we placed the seed into a little paper cup and put the inoculum below the seed. So we've got a layer of the pathogen inoculum just below where we would plant the seed. So the seed would get a little bit of a chance to start germinating before it came into contact with that particular pathogen. And then we're able to take them out. The vermiculite's really nice. We can wash that off and then scan, look at root area and root mass and other parameters. So here's some data then from that seed rot assay. So the higher we are on the bar here, the more disease we have. And I've got some graphics along the top to try and express that. So the far right-hand side here, all those seed are essentially not germinated. And, we've got essentially complete death there. As opposed to the far left-hand side where we've got our control points here and an example of that. And then some of these intermediate reactions we're seeing. So we did this assay at two different temperatures. It was a reasonably rapid assay, so we're able to accommodate a couple of different temperatures. So we chose 13 degrees Celsius, which is 55 degrees Fahrenheit, probably representative of some of the planting temperatures that we use up here in the north, and then 20 degrees Celsius, which might be more representative of some of those southern planting temperatures or, some sort of replant situation. Phytophthora sojae, like I said, was not very aggressive in this particular assay. It's certainly like the warm temperatures. So at that 68 degrees Fahrenheit, it was a lot more aggressive than at 55. But overall, very low pathogenicity in this seed rot assay compared to a lot of those Pythium species. And so you can see some pretty big separation between the warm temperature and cool temperature pathogenicity data. And just by the way, too, we use 3 different isolates for each of these species to get these numbers. So we've got a little bit of spread, but that's across 3 different isolates as well. So I was pretty happy with this data set. But you can see some species really do prefer warm versus cold temperatures, and there's opposite as well. So this is a good example of the opposite with this particular group of species here, the Pythium upapillum included, is preferring the cooler temperature as opposed to the warm temperature. And down here, we've got Pythium aphanidomatum and Pythium Phytophthora sansamiana that certainly prefer these warm temperatures as opposed to these cooler temperatures, being much more aggressive at those temperatures. And then this is the seedling root right assay. So this scale is different. This is looking at the amount of root area. So the higher on this chart, the healthier the plant is. So our controls are actually down here at the far right-hand side of this graphic, where we've got a lot of root growth. Okay, and all of these are not statistically different to that untreated check. But then we've got a lot of these other species that are doing considerable damage in this particular type of assay, including with this blue arrow here, we're looking at the Phytophthora sojae. So it appears, I mean, from this work that Phytophthora sojae appears to be far more of a seedling damaging organism as opposed to a seed rotter. And we've sort of characterized them as either rotting seed, doing damage on roots, or potentially both. And so we've got these sort of categories that we can work with, which is helpful and interesting when we start to think about either breeding for resistance or control, be it biological or chemical. And here's an abundance of the top 8 pathogenic oomycete species. So the most abundant species across both of those years of the survey, and those that were pathogenic, not including those that were not pathogenic. And honestly, when we set out, I was a little worried that we might get very different information across states because we're relying on different people to do things right together, and that's always a challenge. But I was pleasantly surprised that we tend to see these sort of similar groupings. So across Michigan, Indiana, and Illinois, Pythium sylvaticum tended to be the most abundant species. So when we look at this statistically too, we see that there is grouping by geography. So the different geographies grouped together, and I guess is pretty clearly abundant down here as well, where we've got Pythium irregulare being the most abundant in Arkansas and across in Kansas too. Okay, so I was pretty happy about that, and I mean this also points to the potential for prescriptive management. Certainly we're a ways from that yet because there's a whole bunch of species out there, but it certainly highlights that there are some regional differences, and we might be able to take advantage of that in the future to be more precise with the particular treatments that we're implementing. So we had this fantastic information in terms of which species were there and what disease that they were causing. The next step that we really wanted to do is look at their fungicide sensitivity across all these different oomycete species, these 82 different species. So that the traditional sort of poison plate that we use, so we'll put the pathogen on a plate that's amended with that fungicide treatment and typically measure the radial growth. That's fine and it works really well. It's our gold standard. But it would be a lot of work to do all of these different species that we had across the different chemistries. So a PhD student in my lab, Zach, developed this high throughput fungicide sensitivity assay. And so what he did is grew up the oomycete species on a very sort of soft agar medium, and then macerated that by just pushing it through a syringe a couple of times to basically grind up the hyphae. And then he was able to use a multi-channel pipette to pipette that into a 96 well plate. And then what we were doing was using optical density to measure how much growth there was in these different fungicide treatments that we had in this liquid broth that we were growing them in. And then there's some other statistics there to look at signal versus noise, this Z factor that's used in a lot of pharmaceutical screens. And so Zach implemented that as a data control, quality control parameter. So here's just an image of what that might look like then. So 2 different oomycete isolates with 3 replicate wells that we have here. No mefanoxiam in the top row there, so we can see really good growth of that oomycete across all of those 2 isolates and the 3 replicate wells. But 1 part per million mefenoxam, this particular isolate is resistant at that level, or insensitive, so that 1 ppm of mefenoxam, so you can see that growth. But this other isolate was sensitive, and so there's no, you can't see any mycelial growth in there. And the same at 100 ppm, both of those isolates were sensitive at 100 ppm. So that's a visual of how it works. We're using optical density to actually give us an actual value on that. But the beauty of that is that we're able to screen through our panel of 82 different species and start asking some questions about fungicide sensitivity. So this is a phylogeny of different oomycete species and how they're related to one another. And what we've mapped on here is either mefenoxam sensitivity or ethoboxam sensitivity. So the darker the coloring here, the more insensitive those isolates or those clades are to the mefenoxam or the fungicide treatment. So you can see across the spectrum, mefenoxam was quite effective. We really didn't see a great deal of insensitivity. There's maybe 1 or 2 species where we're starting to see, some sort of increased level of insensitivity, but by and large, we didn't really find any. Incidentally, in terms of looking for isolates that were what we would deem resistant to mefenoxam, there's a couple of, I think it's Pythium ultimum out of Washington State from chickpeas that were very resistant. And so we use that as one of our checks, but we weren't able to find any in that survey of Pythium species from across the soybean production area. Ethoboxam is a different story, and Valent certainly knew about this, but we explored that for them further and just identified particular clades that were essentially resistant to that ethoboxam treatment. So what was really interesting too is that that was a new product, so there wasn't like that product had been out and we built up resistance to this. We actually found out that ethoboxam, that particular insensitivity to ethoboxam, is something that's even in the evolution of these oomycete species. So there might be similar sorts of compounds that are produced by plants that have a similar sort of binding as this ethoboxam product does. And so that might be why we see these particular groups that have resistance to ethoboxam. And that's fine. Ethoboxam is still a really good product and certainly pairs very, very well with mefenoxam, particularly in cases where we have some of these species that might be present, like Ephanidamatum, that we might be trying to control. So I think really helpful information to know in terms of, what is happening out in the field, because it's always a little bit tricky to know exactly what's happening out in the field without a lot of work. So from that disease seedling survey, we identified more than 40 species capable of causing disease, a total of 82 species in total that were recovered, but not all of them pathogenic. Temperature was certainly shown to be, affecting which species can be problematic. And so that can be certainly thought about in terms of any, predicting risk in the future. Like, are we having a cold snap come through right after planting? Like, what is that going to mean for the potential Pythium species that we see on those seed or seedlings? And I guess the take home for a farmer would be that, combining plant resistance, as always, is a really smart thing to do, and then combining that with an effective seed treatment. And then ethoboxam is a really good partner in with the mefenoxam to make sure we've got control of everything. All right, so this is some more recent work that Austin, current PhD student in the lab, is doing, and Ann Dorrance at Ohio State and Alison Robertson at Iowa State have been doing a lot of this work as well. So we're getting a pretty good idea of what races are happening across the US. We hadn't done a survey like this in Michigan for about 20 years. And so we were really keen to figure out what pathotypes or races of Phytophthora were present. And then also to start looking at the fungicide sensitivity. Is there any fungicide resistance developing? Mefenoxam's been around since about 1977, so it's been out there for a while. Just in case you don't know what might be involved in a Phytophthora sojei isolate collection. So Phytophthora sojei is pretty slow growing, and that sort of makes it hard to isolate or more challenging than say some of the Pythium species that we work with. So it's a pretty involved process that we have to go through. So Austin solicited a whole bunch of soil samples from across Michigan. We go through this flooding and then drying process and incubation, and then we plant them out with susceptible soybean seed, cover that with a bit of vermiculite, let them grow up, and then we do isolations from those that get infected with the pathogen. So we're baiting out the Phytophthora sojei. And then we end up hopefully with a nice pure culture of Phytophthora sojei that doesn't have bacteria or anything else on it. And then we can ask questions. So what race or pathotype is it? And this is how some of the pathotyping is done. So we use differential cultivars. So each soybean cultivar will contain a single RPS gene, or a gene that's resistant to Phytophthora sojei. That's what the designation sort of comes from. And that way we can determine, which genes are no longer effective and which RPS genes still have efficacy for controlling the disease. So we use this method of using a syringe to make a little slit and then pushing out some of that agar slurry that contains the Phytophthora sojei onto that cotyledon, and then wrapping that, or covering that actually, to maintain humidity, and then looking for plant death. So here's Austin, hopefully not sticking himself in the finger there, but a cartoon of what we might see. So here's the 14 differentials and their response to this particular Phytophthora sojei isolate. So we're always looking for the RPS gene, the control that has no RPS genes. This particular cultivar to always die. If it doesn't die, then we know we have something wrong with the assay, because there's no resistance gene in that particular line. But then all of these others, they contain these different RPS genes that are indicated, such as RPS3A. And so we inoculated that, but it doesn't die. So we know that resistance gene is still effective against this particular Phytophthora isolate that we're inoculating with, and so on. So the blue lines here indicate, from the 2001 study that was published more than 20 years ago now, which RPS genes were effective. So, or ineffective, I should say. So the higher the line here, the less effective those genes are. So in this instance, where we have no RPS gene in our check here, 100% of those isolates for the 2001 study are causing disease. So it's killing all of those plants. The 1B was effective, 1K was effective. There's still a few isolates that are capable of causing disease, but below our threshold of 30 to 40%. So we would still say, by and large, the RPS 1B and the RPS 1K were effective 20 years ago. So what we're showing here in red is the current study that Austin's getting wrapped up right now. And you can see that there's been a pretty dramatic shift. So unfortunately, the RPS 1B is no longer effective. So 90% of the isolates that we looked at in Michigan are now capable of causing disease on that RPS 1B gene. The same with 1C and 1K, a couple of genes that have been heavily relied upon and are deployed in a lot of the germplasm that's out there. Interestingly, 3A was effective 20 years ago and is still quite effective at the moment. Then 3C and 4, getting marginal, but there. So there's been a pretty big shift in fungicide in Phytophthora sojei isolates and their ability to overcome these different genes. And so that's really important from a breeding perspective, either making sure we've got those effective genes into the different cultivars that we're deploying, or also relying on that a field tolerance or partial resistance. And I know Ann Dorrance is a really big proponent of that, making sure we've got that partial resistance or field tolerance. But the interesting thing there is, if you're going to use partial resistance, you typically need to pair that with a seed treatment, because very often those seedlings don't always exhibit those partial resistance at the early plant stages. So a seed treatment can really help protect until that plant is then exhibiting better partial resistance to Phytophthora. And so that's the next part of the work that Austin's been doing, looking at these different products. I need to get permission to share the oxythiopropyllin at the moment. But what we're seeing overall is that there's pretty good efficacy. We don't really see any case in Michigan, at least, for resistance to mefenoxam And again, that ethoboxam product was quite good at managing the Phytophthora sojei isolates that we looked at. We also looked at pyraclostrobin, and we're looking at azoxystrobin at the moment too. There's more variation there. I mean, they've got a wider spectrum of activity as well, so they also do provide activity against true fungi too. So some action points in terms of Phytophthora management. You know, unfortunately, some of these RPS genes that we've relied on for a long time, like 1C and 1K, are certainly becoming less effective. And that's not only true in Michigan, but I know other parts of the country as well. So, and we heard as well that some of the RPS3A genes that were deployed this last year, or those cultivars that had that 3A gene in it, did quite well in areas where there was some suspected Phytophthora pressure in the past. So it's sort of checking out in terms of anecdotal evidence as well. So from a grower perspective, you know, if you're dealing with Phytophthora, not only try and look for those RPS genes if you can find them that are effective, but also be sure to look for a good field tolerance score, and then making sure we're pairing that with a good effective seed treatment, mefenoxam, metalaxyl, limacina, and Intego. All right, so now I'm going to transition a little bit more along the risk prediction sort of work that we've been focusing on with soybean sudden death syndrome. And there's a number of people that have worked on this over the years now. So I'll just put their photos up here. So I think most of you are probably pretty familiar with Fusarium virguliforme lifecycle. Just a quick reminder that there's essentially two parts to this. So it infects, you know, as chlamydospores or hanging out on some of the organic matter in the soil, infects those plants pretty early in the season. And it causes root rot as well as the foliar symptoms. So the fusarium is only infecting the roots. It doesn't move up into the foliage, but it does produce a toxin that gets translocated up into the foliage. Occasionally we'll see it in seedlings, maybe at the V3 stage where we have a leaflet, and then we may not see any disease for the rest of the cropping cycle, just depending on how things are proceeding. Or it certainly might turn pretty severe, and we see very heavy SDS symptoms and defoliation, pod abortion, and all that. So just a reminder, it's not only that, I think we're also going to be dealing with yield loss, not only when we see it in terms of the foliar symptomology, but there's a root rot component going on as well. And that's what we're going to talk about right now. So Jay in my lab developed this real-time PCR assay. We spent quite a bit of effort trying to make sure it was a pretty solid assay and didn't cross-react and all that sort of work. So developed this assay that we could then use to ask questions and as a prediction tool as well. And so the way that we use this to look at different colonization of soybean plants was to take these soybean roots, grind them, dry them, grind them, do DNA extraction, and use a real-time PCR instrument to ask how much Fusarium virguliforme DNA is in that soybean root as a way of measuring how much that SDS fungus is present in that root system. So we did a study where we looked at both what we would be considering, susceptible varieties and those that were moderately resistant or resistant. So those that perhaps would not produce any foliar symptoms, and some of those that would produce foliar symptomology. So we learned a couple of different things from doing this particular trial. We learned that, well, first off the bat, the amount of Fusarium virguliforme in that root system, we can detect it as early as V3, and then it continues to increase throughout the season, and even post-harvest, we see a bit of an increase at times, depending on when we capture that senescence. There always appear to be a little bit of a dip, and we saw this across a couple of years. That might be something related to root growth. We're not really too sure, but in general, we certainly see an increase or an increase in the concentration of Fusarium virguliforme in those root systems. And this is just taking the entire root system and grinding them up. There's certainly other studies that could be done now to look at taproot versus lateral roots, different positions of lateral roots. There's all sorts of questions that could still be asked, but this is what we saw, this general increase. The other really interesting thing that I thought was really quite remarkable is that even in some of these more resistant lines, so this one in yellow and this one in blue, that never showed any foliar symptomology, the amount of fusarium virguliforme in that root system increased over time, very similar to those that are susceptible and developed a lot of foliar symptomology. And most of the time, there was no statistical difference. So in terms of management, it's great that farmers are using varieties that have better resistance packages. That's fantastic. But I think people need to be very careful that they don't go ahead and use a resistant variety, don't see any foliar symptoms, sort of forget about SDS, come back, plant something that might be more susceptible, and then suddenly get hit with this disease and then wonder where the hell it came from. So, you know, unfortunately, and we also know from other work that the SDS fungus is quite capable of surviving on corn debris. It infects root systems, but corn root systems, but to a much less extent, but it's able to survive on that corn debris. So it's a difficult pathogen to manage in terms of those crop rotations. All right. So anyway, I thought that was really interesting, and again underlines that point I was trying to make about we might be losing yield even if we don't see foliar symptoms, much like soybean cyst nematode, right? So there's some root rot going on too. So this brings me to the risk prediction tool that we've been working on for a few years now. Mitch did some of this work with Jay and others in the lab. So what we did here was essentially looked at our location with heavy SDS pressure as a proof of concept study across a couple of years. Can we predict where in that field we're going to develop hotspots of SDS and yield loss based on this real-time PCR assay? So just detecting how much fusarium virguliforme DNA, will that be a predictor? And so this is an overhead view of what Mitch did. So we basically just gridded out a field and took all these point samples and observations and soil samples from across those locations. We looked at all these different correlations as well. We also looked at soybean cyst nematode, and we saw that soybean cyst nematode also correlated in a positive way with the amount of SDS that develops. So down here, we see the blue is a positive and the red is a negative correlation. So for example, down here, we can see that the at-planting cysts and eggs positively correlate with the amount of SDS that develops in this area that we looked at. And that's interesting as well, because there's certainly been a lot of studies, some that show that there is a relationship, others that show that there is no relationship. So this is another piece of evidence pointing in the direction that there is some sort of relationship. We've been trying to do more work to understand what that is exactly too. And then other components like at-planting fusarium virguliforme, how does that correlate to yield? It's negatively correlated. So if we see a lot of fusarium virguliforme, we know that's going to be negatively correlated to yield, and positively correlated to the amount of soybean sudden death syndrome that we see develop. So here's a map of that, just looking at it from above. So the scale there is in meters. We try to do this for colorblind people. I think I still need to flip the scale yet again, because it's not intuitive. So the lighter the color, the more of that particular organism we have. So here we're seeing more fusarium virguliforme at the top of the 2014 map and at the bottom of the map in 2015. And we went ahead and also mapped in the number of soybean cyst nematode eggs. So we had some hot spots in various parts of the field. And then from those samples that were taken at planting, we were trying to predict how much SDS develops, how much foliar disease develops. And so we see a correlation where we see a high amount of fusarium virguliforme, we see that high amount of SDS develop, and a loss in yield across years. So again, high amounts of SDS down here, higher amounts of the fusarium virguliforme fungus, higher amounts of SDS and lower yield in those particular areas. So we saw this pretty tight correlation. And like I just mentioned too, that the soybean cyst nematode also contributed, of course, to yield loss, but also to SDS foliar symptomology. And so this is a schematic that, or a sort of example of something that Mitch put together here, how we might start to use this. So we're probably going to look at a number of different factors. Like, what is the field history? Have we ever had SDS in there or not? And that might contribute something towards this score. And then, of course, using our virguliforme assay on that soil sample, how much do we detect and how does that predict whether we're going to be in a low, middle or high risk category for SDS? And of course, the number of soybean cyst nematode. If we've got really low numbers of soybean cyst nematodes, it's not going to contribute as much as if we have high numbers of soybean cyst nematodes in terms of that SDS and potentially yield loss too. So in Michigan, we've the approach now that we feel very confident that we've got an assay that works, at least in that one location. We've been doing it across multiple locations in Michigan for the last 4 years now. And in 2020, we teamed up with a lot of pathologists from across the region. I think we've got about 60 different sites that we're trying to look at this relationship to basically create larger data sets to improve the risk prediction. So in terms of if you're a farmer, in terms of action management, then certainly use resistant varieties. Again, it's going to be our first point of recommendation when trying to manage any disease. Make sure you're using the best varieties in terms of resistance that you can. And we've also been looking lately at the various seed treatments, Olivo or Saltro, both appearing to be quite good. We've looked at Olivo for many, many years, Saltro just recently, and certainly more profitable in locations with heavy SDS pressure. And again, that's where we want to implement this assay to figure out, like, should we be using that seed treatment, which is an added cost, or not? And so if we can predict where the SDS pressure is going to be, then we can make that recommendation that, yes, go ahead and use Olivo or Saltro as a treatment. And of course, always test and manage for soybean cyst nematode. That's also a yield robber. And there's been some work out of Iowa State to show that extended crop rotations might certainly do reduce SDS pressure. I guess the question is now, do these cover crops, do they have potential to fit into that? If we can't get away from that corn soy rotation, can we use cover crops to increase diversity and reduce SDS pressure? So just my last slide. So hopefully what you've gotten from this is sort of a sense of what we're trying to do in the lab in terms of understanding soybean seedling diseases. It's extremely challenging to get a call out with a grower and then try and figure out exactly what's going on, because it's incredibly involved to do isolations and figure out what's there. So we've been developing diagnostics and, again, trying to predict risk and figure out which species are actually, major players and which ones aren't, and which ones have, fungicide susceptibility. So these tools that we've been developing hopefully will improve screening of new fungicides as well, and certainly monitoring for any fungicide resistance issues that might be starting to pop up. And unfortunately, pathogens continue to adapt, right? So you saw that with our Phytophthora sojae and the different races or pathotypes that we're seeing. And our end goal here is really working towards disease risk prediction to return or improve that return on investment. And with that, that's my last slide. And I just want to thank everyone that technicians as well that do an incredible job on the program. So be happy to take questions or comments even.
All right, Marty, thank you. And just to introduce myself, guys, I'm Kirsten Wise. I am an extension plant pathologist at the University of Kentucky. And I'm just going to have some discussion with Marty now, just a little bit about some of the things he talked about and some of the questions that you guys are putting in on the Q&A, and also some of the questions that just in general, Marty and I get, and sometimes we get for each other. So I've known Marty a long time, so I'm going to ask you some really hard questions, Marty.
Great.
So first of all, I do want to, there's one that just came in on the Q&A, so we'll address that first before we get to some of mine. We'll break you in. But one of the questions was, they said, your SDS spatial data was interesting from a research perspective. Do you anticipate farmers wanting to understand their risk in this much granular detail if they will not even scout their own fields?
That scouting issue, right? Like that's something I think from the extension standpoint, we're like, did you really get out of the truck? Like, anyway, so yeah, from a granular detail, no, I'm not interested in that either. Like, we did that granular study as a proof of concept. Can we actually predict in a single field where there's going to be hot spots or not? It was the easiest way just to see if the tool would work. Now we know that the tool works. We're scaling up. So we're just collecting soil samples from different fields to see if we can predict that risk. But I really don't care about, sub-meter accuracy of SDS risk prediction. That's not of interest at all. there's certainly potential for prescription management, right? If we can identify from satellite imagery or drone imagery that, oh, you've got a hotspot of STS in these, this compacted area of the field, that's maybe where you want to drop that treated seed versus other areas. Yes, for sure. But I'd much rather have a, field by field sort of approach. And maybe with some, if they're huge fields, maybe breaking them into quadrants or something, but not that granular approach. It's just to demonstrate the tool.
Right. Yep, that makes sense. I was going to ask you you talk a lot about some of the seed treatments, and that's sort of been our gold standard, particularly for how we're managing things like Pythium, right? And even to some extent Phytophthora, if we don't have good field tolerance, or even if we're using less resistant varieties. But one question I often get asked is about in furrow fungicide, right? So there's more products able to put in furrow, and some of those products are going to have maybe some strains of bacillus, or maybe some of the older QOIs go in furrow along with that seed treatment, and it's promoted to enhance that seedling disease control. So have you looked at any of those?
Yeah, we have. I'm forgetting the name of the product now, but it was an FMC product that had that bacillus species in there. And I mean, typically, the biologicals unfortunately don't tend to fare as well, right? But we had an instance where the biological in that treatment, because we had it with and without the biological, and the biological really seemed to be for that particular treatment. I think there was an insecticide in there too. Anyway, but yeah, the biological certainly did a really good job there. And we've certainly had other industry trials where, yeah, the biologicals have been surprising. There's been all this venture capital poured into like biologicals, right? Everyone's super geeked about it again because we can actually sequence the soil and maybe get a better understanding of what's there. But yeah, surprisingly, there's been some pretty positive responses. So yeah, that's really cool for sure.
Yeah, and I would say in our work that we've done, we've seen some of the same responses where some of the biologicals have been interesting when we're putting it in furrow. We've seen a little bit better response when we're using it in cooler, wetter soils, soils that are more prone to some of our seedling blights. Have you seen that as well?
I would say yes. Yeah, I should have pulled a specific slide to answer that question, but yeah, I would say so. Yeah, for sure. I mean, I think anytime you're under greater stress, right, then just like everything else, foliar, fungicides, whatever it is, you're more likely to see a response. So I would say so, particularly if those biologicals are helping to protect from those stresses. Yeah, those pathogen stresses. Yeah.
It would also be interesting. I mean, you had that really nice data set where you're looking at that species composition, right?
Right.
And I think that makes maybe a big difference is that I don't think we've had tested the in-furrow fungicides enough to really know how it's going to play out against some of those different species. And so that'd be something.
Yeah. There's so many questions, right? That's part of the.
I mean, my job is just to put stuff on your plate, Marty.
So I guess some of the approach we've taken there too, a few years ago, we certainly were like making this cocktail of different pathogens and then testing a treatment. We've shied away from that because then if you get a response or you don't, it's like, well, okay, great, but which species was driving that? So the approach we've taken lately is to reduce typically one pathogen per trial, right? Particularly those very aggressive Pythium species, and then see what sort of control we can get with those fungicides or biologicals in furrow or seed treatments. And that's, at least we can build on that a little bit more, right? As opposed to like, well, I don't know what it was.
Yeah, that can be really frustrating when you don't know what actually is causing your response, right?
Very much. Yeah.
So I was also going to ask, so whenever I talk about, the new seed treatments, putting in furrow stuff in, I always get the question about, what some of these different treatments are going to do to soil beneficials, cover crops, and there's sort of a lot of interest in how we can build up, beneficial organisms in our soils and make our healthier. And I don't know, what do you think about that?
Okay, so I'll answer it in the flip first, in the flip side of things. So, so we've looked at seed treatments in terms of what is their impact on even the pathogens that we're seeing, and what is the risk for resistance to fungicide development. And the risk seems to be pretty low because you're putting a very small amount of chemistry in a very small area, right? So the risk is low. Typically from a seed treatment and probably, well, even in furrow, I'd say you're not really changing the community in the soil a great deal. But you certainly may be influencing what's growing on the roots. And it's actually been frustratingly difficult to try and find. We can find differences, but it's been harder than what you might think. But yeah, from the beneficial, absolutely. I mean, that's I question why we see some of those negative responses that I was showing earlier, right? And I know as pathologists, when we get together, we talk about this from time to time. It's like, well, what else are we doing, right? If we knocked out something that's allowing the pathogen to come in after the fact and then do more damage than what it should be doing. The answer is I don't think we know. I don't think there's any good studies out there looking at that, and some of those mycorrhizal species are pretty hard to work with too. So that's the other challenge. Like some of them are really hard to culture. They need living plants. Like that's certainly something that I think would be really cool to start looking at in more detail. But it's challenging work for sure.
I always try to make that point when I get asked that question, is that it's a really important and interesting question, but it's such a difficult question to study, right?
Yeah.
Also because, your populations, your native microbial populations in Michigan are going to be really different than our native microbial population in Kentucky, and even from field to field.
Yeah, definitely.
And so that out is really hard.
Yeah, and that's what I was trying to point out too with that. Yeah, I guess I had well, I shouldn't call him naive, but a new graduate student, right, is like, oh, well, seed treatments are useless, right? Look at my data. Like, no, you can't say that. My goodness, look at what's happening at Allegan. 15 bushel difference to the untreated at 160,000 seeds per acre, and not at a crazy April planting, but a May planting, right? So yeah, there's a lot of value to seed treatments, but yeah, I mean, if we could figure those relationships out better, maybe we could improve those returns on investment even more, right? And that's what is where we're trying to head towards.
Yep, I agree. So I just want to ask just a few just sort of general things. I know that sometimes people maybe don't realize, like, why is it important to do this type of research? You're looking at things, not just management options, but we're really fundamental stuff about the pathogens here. And why is that important to do?
Well, I think I could have approached this presentation just by looking at product performance, right? And that's great. And it's really exciting. And, especially a lot of these new things coming up, you can give a new talk, every season easily, right? There's always new combinations and things, but it doesn't get at the underlying, like what's really going on, right? So I feel like we keep hitting our heads against the wall if that's all we're doing. So some more fundamental understanding of what's really going on can actually lead to some of those advances. Like, oh, okay, we know that ethopoxam doesn't work very well against Aphanidomatum. So in locations with heavy Aphanidomatum, make sure you use that Intego Suite, I think is what it's called, right? Where you've got mefanoxam in the blend as well. And I'm sure we'll probably see similar results for other chemistries we're currently screening too. So I think that's why it's really important. I think there's a pretty good return on the investment checkoff dollars that come into this. And we certainly get asked by industry all the time for this information. Can you share those slides yet? They want to know what's out there too, of course, because they're going to be screening in-house or whatnot, or working with us to screen specific products against particular issues, right? So I think it's really, really important work.
Right. Yeah. I agree, because it's hard to manage things if you don't know what you have.
Right. Absolutely. Yeah. Yep.
Really understanding, those details about our pathogens, I think, in the future, it's really going to help us to manage these seedling diseases a lot better.
Yeah, I agree.
Yeah. Well, I will turn it back to Kayla. Kayla, are there any more questions? Oh, we got one that came just now. It says, Marty, did your work looking at Pythium species in the north central states find any species specific rule? For example, in this field, Pythium species 1 is most prevalent, meaning that seed disease is most likely to occur when the spring is warmer and more moist, and this particular seed treatment AI would provide the best protection.
I would say we didn't get quite to the end of that goal, but yeah, in general, yes, in general. But yeah, again, that's so the work that I showed you, and Kirsten was a part of that too. Like, it was an incredible effort to, those couple of summary slides that I showed you were a couple of years worth of work, right? So that's the problem. We don't have really rapid tools for the most part. We're developing them, but we don't have the predictive tools where they need to be for that sort of work to happen. But that's certainly where we want to get to. And I think the closest instance is this SDS risk prediction tool that I talked about. I'm quietly confident. I want to make sure that it works before we across multiple fields before saying this is definitely something that you guys should be using. But I'm really excited about that, and that's what we're really focused on at this point in time. But yeah, we're trying to pull along those Pythium and phytophthora risk prediction components as well. Hopefully it answers that question. Yeah.
All right. Well, I will turn it back over to Kayla. If there's any other questions, that'd be great to answer them. But if not, Marty, thank you so much. This was really great, and we appreciate you being on here to chat with us today.
Yep. Thanks for having me, everybody.
Yes, thank you so much. This was the first in the Spring Crop Protection Network webinar series, so there's more to come. The next one is actually on soybean taproot decline diagnosis and management, and that's going to be with LSU Ag Center Associate Professor of Plant Pathology Trey Price. So that's coming up next month, and you can see all of the webinars that are coming up with the bit.ly link that I put in the chat box, so you can see that. And I want to say thanks to Marty Chilvers and Kirsten Wise for great content today. Appreciate you letting the Southern IPM Center host this. We love being able to spread this knowledge as far as we can. So thank you for that.
Thank you, Kayla. It was great.
Thanks, Kayla. Thanks, Kirsten.